Good morning, everyone. A very warm welcome to all the attendees present here in person and those joining us virtually. We are delighted to have you for today's Physiology Alumni Lecture Series, PALS, which is part of the Silver Jubilee celebrations of the MD Physiology program at Sri Ramachandra Medical College and Research Institute, Chennai. MD Postgraduate program commenced with 3 postgraduates in the year 2005, and so far, 58 MD Physiology graduates have come out with flying colors. Currently, 10 postgraduates are undergoing the program. This monthly hybrid lecture initiative is designed to bring together our esteemed alumni, students, and faculty to share knowledge, inspire academic growth, and create a strong community of physiologists designed to benefit the current postgraduates and physiology enthusiasts of an institution and nationwide. This program shall serve as a bridge between the past and present, fostering inspiration, collaboration, and academic excellence between the alumni of the Department of Physiology. By creating opportunities for alumni to contribute to the academic growth of their alma mater, PALS embodies the spirit of lifelong learning and professional development. Join us in celebrating this milestone and enriching the future of physiology education. It's a privilege to have Dr. K.V. Somasundaram Sir, Academic Advisor in our midst. I kindly request Sir to address the gathering.
The topic is as old as glucose metabolism, obesity, diabetes, the disease. I ready for the dissertation quickly, so I noticed from the time mankind was started registering their civilization, disorders related to sugar were not known as carbohydrate or insulin. Disorders due to sugar were always there. They did not realize it was a disease or anything about it. They realized eating sugar for some people was bad. So they could only call. Starting from that, history of diabetes is as old as human civilization. Starting from that, I'm reminded of what I read in newspaper. How many of you remember it? I don't know how many have time to read a newspaper in the morning. Today in India, they have permitted a new drug officially, which is useful for treatment of obesity and diabetes, the parental drug. So it's a very good day for sugar, diabetes. Thank you for coming.
I request Sir to felicitate Dr. Preetha Paul ma'am as a token of love. It's a great privilege to welcome Dr. Preetha Paul, Professor and Head of Physiology, Panimalar Medical College, Hospital and Research Institute, to lead today's session. Before we proceed with the lecture, let us take a moment to learn more about her professional journey and her significant contributions to the field. I now invite Dr. Meenakshi ma'am to introduce Dr. Preetha Paul ma'am.
Thank you, Jimmy. A happy morning and hearty welcome to everyone who joined us in both offline and virtual mode for our very own PALS 3 Physiology Alumni Lecture Series. We are thrilled to have you all for this insightful session on glucose homeostasis, an essential topic in metabolic health and disease management. It is my privilege to introduce our distinguished speaker of the day, Dr. Preetha Paul, a proud alumnus of Sri Ramachandra Medical College and Research Institute, currently the professor and head in the Department of Physiology at Panimalar Medical College Hospital and Research Institute, Chennai. In Standard 12, Preetha Paul scored state first in Tamil Nadu, for which she received Arinjaranna Scholarship. Dr. Preetha Paul holds an MBBS and an MD Physiology. Ma'am is the recipient of Merit Award for Jawaharlal Nehru Memorial Fund for securing first position in MD Physiology examination. Ma'am has completed her advanced course in medical education and basic course in biomedical research. Beyond academia, Dr. Preetha Paul is a member and resource person, Medical Education Unit and Anti-Ragging Committee at Panimala Medical College. She is also a life member of Association of Physiologists of Tamil Nadu, Indian Medical Association, Christian Medical Association of India. Her work exemplifies the seamless integration of research expertise with 13 impactful publications and educational leadership. Without further ado, please join me in welcoming Dr. Preetha Paul to guide us through the fascinating world of glucose homeostasis. Welcome, ma'am.
I kindly request the participants to please fill the pre-test form posted in the chat box.
We can just do the height there and get it invited.
ಅದು ಓಕೆ ಮ್ಯಾಮ್.
Yeah. Yeah. Is that? Okay.
Thank you so much.
So thank you, Meenakshi, for the introduction, and a very good morning to everybody present here and those who've logged in online as well. It's a pleasure and an honor to be invited here to be part of PALS and to deliver this lecture on glucose homeostasis today. And I thank Prisla ma'am for the opportunity, and Dr. Pratibha, thank you so very much. And I'm very happy to see some friends here and some of my teachers also. Really happy to be here. So I'll move on to the talk now. Thank you. A heavy meal, perhaps just a light meal, even with fasting. Blood glucose levels are maintained constantly within physiological levels of about 80 to 140, despite variations in intake. So why should glucose levels be maintained? It's because, importantly, the only substrate the brain can use for fuel is glucose. And not just the brain, also the retina and the germinal epithelium of the gonads. So if glucose levels are low, what we call hypoglycemia at values less than 80, what happens is the brain will not receive substrate for fuel. And the thing with hypoglycemia is that, as you can see, at levels of about 60, cognitive dysfunction sets in, and if not corrected, will progress to coma, convulsions, and even death. And if glucose levels are high, what is the problem? Levels more than 140 are called hypoglycemia, and that leads to cellular dehydration and tissue injury in the long term. That is the importance of glucose homeostasis, which is our topic for today. And in this session, we are going to be talking about mechanisms of blood glucose regulation, influences on these mechanisms. So this is relatively newer, and I'm going to focus on a lot of recent advances here. And then we'll take a look at the pathophysiology of diabetes, and we are done. So a little history before we go into the regulation. Way back in 1869, pancreatic islets were discovered by Langerhans, and in 1921, insulin was discovered by Banting and Best. So at that time, the focus was on the monohormonal model of glucose homeostasis. Only insulin was considered to be a player. About 30 years later, glucagon came into the picture, and then came the bihormonal model of glucose homeostasis. Insulin and glucagon alone were considered to be the players. Later, gut hormones were discovered, that was in 1970s, and of late, more after 2010, the role of CNS is coming into the picture. So now a lot of information on this particular aspect has been unraveled. So current thinking is that blood glucose is regulated by these three mechanisms: metabolic, hormonal, and neural. So before we go into details of this, I would like to mention 2 points. How is glucose added to plasma? Glucose is added from the food that we eat through the intestine, from breakdown of glycogen in the body, which is called glycogenolysis, and from the process of gluconeogenesis, which is when glucose is formed from non-carbohydrate sources. How is glucose removed from the plasma? Again, by 3 means. One is storage in the muscle as glycogen, storage in the liver as glycogen, this is glycogenesis, and utilization by tissues for energy production, which is what we mean by peripheral utilization. Now, metabolic regulation is by the liver. So when glucose is high in the blood, the liver takes it up and stores it. And when glucose is low, the liver releases it. This system can maintain plasma glucose for about 8 to 12 hours. And this is what operates when we sleep. So that is why the liver is called the glucose buffering system. So what are the mechanisms that operate in the liver? So when glucose is in plenty, the process that takes place is called glycogenesis. So you can see here, glucose entering the cell, it diffuses into the cell, it is taken up by glucokinase. Now glucokinase is the enzyme that is going to temporarily trap glucose inside the liver. And then it is either used up for energy production, you can see it coming down to the pyruvate, or it is stored as glycogen by the action of glycogen synthase. So glucose enters, it's either used or it is stored. And the storage is what is called glycogenesis. This is what happens when glucose is in plenty. Now, when glucose is low, what happens is called glycogenolysis. So here you can see there's glycogen and there are some enzymes acting. The first enzyme you see there, glycogen phosphorylase. Now, this is present in muscle, in liver, and also in brain. Whereas the last enzyme that you see, the glucose 6-phosphatase, this is present in substantial amounts only in the liver. So the last step of glycogenolysis can be done only by the liver, and that is why the liver is responsible for releasing glucose into the circulation. The other process, so that the amount of glucose that the liver can store is quite a large amount as glycogen. It can store about 100 grams, which is 6% of its mass. Now, the other process by which glucose is released into the circulation by the liver is what is called gluconeogenesis. Now, 2 things are needed for this process, the enzymes in the liver, and the other thing will be the substrates to be used. Usually, muscle is catabolized to produce amino acids, which are brought to the liver again for gluconeogenesis. Also, by lipolysis, fat is brought to the liver, and then that's used for gluconeogenesis. Now, these processes that are being done by the liver, they are not independent processes. These are under hormonal control. And that brings me to the hormonal regulation of glucose homeostasis. So I'm going to restrict myself to the main hormones that are players here. So we have pancreatic hormones, we have hormones from the adrenal glands, and also from the pituitary gland. So pancreatic hormones that play a role in glucose homeostasis are insulin and glucagon. From the adrenal medulla, we have epinephrine and norepinephrine. From the adrenal cortex, we have cortisol. And from the pituitary, we have growth hormone. Now, insulin is the hormone that will decrease blood glucose. All the rest will increase blood glucose. Okay, so insulin is the only hormone that's going to decrease blood glucose. All the rest will increase blood glucose. Now we'll take a look at all of these hormones and their actions. Now insulin, you all know, is secreted from the beta cells of the islets of Langerhans. The islets are the endocrine portion of the pancreas, and along with insulin, C-peptide and amylin are also secreted. Now C-peptide is the connecting peptide that is released when proinsulin is cleaved to form insulin. The importance of C-peptide lies in the fact that it is released in equimolar quantities with insulin, so it serves as a marker of endogenous insulin secretion. I'll come to amylin shortly. Before that, we'll talk about insulin. Now, insulin is a peptide hormone which acts through its receptor. So any tissue in the body which has insulin receptor, insulin will produce its actions there. Insulin receptors are densely found in the liver and in fat cells. You can see from the picture, you can see the cell membrane over here. And this transmembrane receptor has alpha and beta subunits. Now the alpha subunit has insulin binding sites, and the beta subunit is coupled to tyrosine kinase, which acts on insulin receptor substrates, brings about phosphorylation and effects. And one important effect of insulin is on glucose transport. So what we see, glucose is a big molecule and it needs a transporter to help it get into the cell. Now in the cytoplasm of insulin-dependent, insulin-sensitive tissues, GLUT4 receptors are present, what you see over here. Okay? And when insulin binds to its receptor, the GLUT4 fuses with the cell membrane so that the GLUTs get inserted into the cell membrane and glucose can enter. Once insulin action is over, it's again internalized, gets back into the cytoplasm. This is what is called cycling of GLUT4 transporters in insulin-sensitive tissues. And GLUT4 is not the only transporter in the body. There are a variety of glucose transporters. In fact, GLUTs 1 to 13 are present. GLUT4 is the only one that is insulin dependent. As you can see, they're present in a variety of tissues in the body. And glucose transporters operate by facilitated diffusion. Which means movement is from high concentration to low concentration through a transport protein, facilitated diffusion. Other than GLUTs, there are other transporters as well. There are the sodium glucose co-transporters. Again, a wide variety found in different parts of the body. These operate by secondary active transport. So from low concentration to high concentration using energy. So insulin-dependent glucose uptake is seen in quite a few tissues in the body, as you can see from the list that I have shown. But there are some tissues where insulin-independent glucose uptake takes place. These tissues do not need insulin to take up glucose, and these include the kidney, the intestine, brain, retina, and RBC. Kidney, intestine, brain, retina, and the RBC. So what is the action of insulin on muscle? Importantly, insulin increases glucose uptake into resting muscle 15-fold. There's this enzyme called hexokinase in the muscle. So glucose enters through GLUT4, is taken up by the hexokinase, phosphorylated, and then as we earlier saw, it's either used or it is stored. It is used by glycolysis to produce energy, or it is stored by the action of glycogen synthase to produce glycogen. And now that there is glucose inside the cell, insulin does the job of switching the fuel use to glucose. So it switches to and increases peripheral utilization of glucose and spares fats. And what is not used is going to be stored as glycogen. So about 3% is stored in muscle. This graph is to show the effect of insulin on uptake of glucose into the muscle cell. So on the x-axis, you can see glucose outside the cell, and on the y-axis, glucose inside the cell. This line shows that though extracellular glucose increases, hardly anything is getting into the cell in the absence of insulin. But take a look at the red line. When insulin is there, you can see a linear relationship. The amount of glucose getting into the cell increases as the amount of extracellular glucose increases. What is the action of insulin on liver? We already saw what happens with the glycogenesis. So uptake is promoted, storage and usage goes on. Increased glucokinase activity, that important enzyme which temporarily traps glucose inside the liver. There's also increased glycogen synthase activity, which brings about the formation of glycogen. And when there is, after the usage and the storage, when there's still excess glucose left, then that is converted into fatty acids and stored in adipose tissue. Insulin also prevents the breakdown of glycogen stores by inactivating liver phosphorylase and also phosphatase, and it inhibits gluconeogenesis by decreasing the enzymes needed and decreasing amino acid release from the muscle. So to put it all together, what are the effects of insulin on glucose metabolism? It increases uptake by the cell, it increases utilization by the cell, it increases storage in muscle and liver cells, and it inhibits glycogenolysis and gluconeogenesis. Thus, it decreases blood glucose. Why are diabetics asked to exercise? How does exercise lower blood sugar? Interesting point over here. So insulin-sensitive tissues contain a population of GLUT4 vesicles that move into the cell membrane in response to exercise and are independent of the action of insulin. It is insulin-sensitive tissue, but even without insulin, post-exercise, it moves into the cell membrane. So this is the reason we ask diabetics to exercise. So a word about amylin. Now, I said it's secreted from the beta cell. It's co-secreted with insulin in a 1:100 ratio. And it has synergistic effect with insulin, so it helps to decrease insulin demand. It suppresses postprandial glucagon secretion, reduces food intake, slows gastric emptying, and inhibits digestive secretion. Nowadays, amylin also is considered an important player in the regulation of glucose. Moving on to the other hormones, there's glucagon, also from the pancreas, from the alpha cells. Its important actions are just two: glycogenolysis and gluconeogenesis. So it breaks down liver glycogen. In fact, it is the most potent hepatic glycogenolytic substance known. It stimulates hepatic phosphorylase, simultaneously inhibits glycogen synthase, and increases gluconeogenesis by increasing the enzymes necessary and providing substrates. Thus, it increases blood glucose. Moving on to the next hormone, which is growth hormone. As we know, it's from the anterior pituitary. It decreases the uptake of glucose, decreases utilization, increases gluconeogenesis and hepatic glucose production. Thus, it increases blood sugar. Similar to growth hormone, cortisol also decreases the uptake and utilization of glucose, and importantly, it is a very, very potent gluconeogenic hormone. In fact, it can increase gluconeogenesis 6 to 10-fold by increasing the enzymes and by increasing substrate delivery to the liver. So these hormones increase blood sugar. And then moving on to the catecholamines, which, as we know, are going to be secreted from the adrenal medulla, especially in situations of fight, flight, or fright. So what do these hormones do? Glycogenesis is decreased, less storage, glycogenolysis is increased, Gluconeogenesis is increased, all this to increase blood sugar. Additionally, it inhibits the insulin secretion, increases glucagon secretion, so that a blood supply to the brain is ensured, and also amplifies the effect of epinephrine by increasing glucagon secretion, also stimulates secretion of ACT so that cortisol is secreted and gluconeogenesis is stimulated. So all these actions tuned to increasing blood sugar. Thus, we see that glucagon, epinephrine, norepinephrine, growth hormone, as well as cortisol, all these hormones increase blood glucose. And this is what is called the hypoglycemia counter response. Hypoglycemia counter response. So, just putting it all together, when blood glucose increases, we have insulin secretion increasing, which increases storage as glycogen, also acts on protein and fat, which is beyond the scope of my talk. So ultimately storage is taking place, usage also, and there is inhibition of glycogenolysis and gluconeogenesis. When blood glucose decreases, then we have the counter hormones coming into play. We have glucagon and epinephrine increasing gluconeogenesis and glycogenolysis, and growth hormone and cortisol increasing gluconeogenesis and decreasing uptake and utilization. Another tidbit now. What is the effect of stress on glucose homeostasis? So we know that in conditions of stress, epi, norepi, and cortisol are going to be secreted. Gluconeogenesis is increased by all these three hormones. Glycogenolysis is going to be increased by epi and norepi, and uptake and utilization are going to be decreased by cortisol. So all these are going to work together to raise blood glucose. Additionally, norepinephrine binds to alpha receptors on beta cells and inhibits insulin secretion. What about chronic psychological stress? What is the effect of that on hormones and on glucose? So this again will stimulate catecholamine and cortisol secretion, leading to elevated blood sugar, which is going to be persistent. So that keeps the person at a high need for insulin. So in the long run, this can lead to insulin resistance and precipitate diabetes. As doctors, we have to remember that when we think of stress, we must remember that illness is stress, surgery is stress, physical stresses are there. So all these stresses will increase blood sugar, and when prolonged, they can precipitate diabetes. So we'll take a look at the influences on pancreatic hormone secretion. We all know that glucose is a prime influence. So when there's going to be glucose in the blood, it is going to trigger and amplify insulin secretion. And when blood glucose is low, it will trigger glucagon secretion. Other nutrients also have an influence on these hormones. So we have amino acids arginine and lysine stimulating insulin secretion. Arginine and alanine stimulate glucagon secretion. Free fatty acids are of two kinds, long chain and short chain. The long chain will stimulate insulin secretion, whereas the short chain will inhibit it. Other hormones also influence insulin secretion. Glucagon, growth hormone, cortisol, and progesterone, estrogen to a lesser extent will increase insulin secretion. So one point we kept in mind is that prolonged secretion of any of these can lead to burnout of the beta cell and increase risk of development of diabetes mellitus. Similarly, when we put patients on high pharmacological doses of any of these hormones, again, precipitation of diabetes must be kept in mind. Interestingly, there is interplay between the pancreatic islets and other organs and tissues. Insulin produces effects on many tissues, and many tissues interact with the islets to modulate insulin secretion. So I have some interesting things to talk about here. So thus we have interaction within the pancreas itself, within the islets. We have islet interaction with the gut. We have it interacting with the adipose tissue, with muscle, with liver, and also with the brain. So we'll take a look at this now under the following headings: the intra-islet axis, gut islet axis, adipocyte myocyte islet axis, liver islet axis, and the brain islet axis. Intra-islet insulin axis. So what is shown in the picture is an islet, And we know there are different kinds of cells in the islets. And generally, the beta cell is more in the center, while the alpha and the delta cells are more to the sides. Now, these cells interact with each other through extracellular spaces and also through gap junctions. Thus, they influence each other's secretion. So we can see here glucagon stimulates somatostatin secretion. Somatostatin is secreted by the D cells of the islets, and pancreatic polypeptide is secreted by the F cells. Somatostatin is generally an inhibitory hormone. Not much is known about the action of pancreatic polypeptide. So glucagon stimulates somatostatin secretion and insulin secretion. Somatostatin inhibits pancreatic polypeptide, also insulin and glucagon, and insulin inhibits glucagon secretion. So this is the intra-islet insulin axis. Coming to the gut islet axis, there are four elements here: the incretins, the decretins, other GI hormones, and lastly, the gut microbiota. Now, the incretins include GLP-1, glucagon-like peptide 1, which is from L cells, and GIP, or glucose-dependent insulinotropic polypeptide, which is from K cells. Now, these incretins are secreted from the enteroendocrine cells of the gut, and oral glucose ingestion stimulates their secretion, and that is what is called incretin effect. The effect is not seen with IV glucose infusion. And the word incretin itself comes from the Latin word increscere, which means to increase. So the prime action of incretins is to potentiate insulin secretion. And it brings about an anticipatory rise in plasma insulin, readying for glucose from the meal. Also has effects on beta cell neogenesis, proliferation, and survival. Additional effects include inhibition of glucagon secretion and suppression of glycogenolysis. Many studies in receptor knockout mice, where the GLP and the GIP receptors have been knocked out, showed defective insulin responses and consequent glucose intolerance. So an incretin deficiency is implicated in the pathogenesis of type 2 diabetes. Moving on to the decretins. I think the term is self-explanatory. We understand that these are molecules that are going to decrease insulin secretion. So neuromedin U and lemostatin are peptide hormones secreted during fasting or food depletion. They're found in highest levels in the upper GI, in the duodenum and jejunum, and they are known to suppress insulin release. In the absence of lemostatin, hyperinsulinemia, hypoglycemia, and obesity were observed. Similarly, with NMU receptor knockdown, the same were reproduced, and there was also diminishing of the insulin suppressing ability. So decretins may represent a novel mechanism for modulating insulin secretion during fasting. Other GI hormones, gastrin, we know it's released from the G cell in the stomach and the duodenum, and CCK, we know it's from the I cells of the duodenum. So these promote beta cell neogenesis and stimulate insulin release, they potentiate insulin release. Postprandial hyperglycemia in type 2 diabetes is due to decreased CCK plasma levels. And a third hormone, ghrelin, which is called the hunger hormone. This is secreted from the stomach. This is an inhibitor of insulin secretion. Also has cytoprotective effect on the beta cells, and it plays an emerging role in glucose homeostasis and body weight control. And coming now to the gut microbiota, which is an emerging area of interest, a lot of interest in gut microbiota now. Aberrant microbiome with alterations in composition, diminished diversity, or even fewer butyrate-producing bacteria. These have been implicated in the initiation or promotion of metabolic disorders like obesity and type 1 and type 2 diabetes. It has been suggested that correction using antibiotics, probiotics, or prebiotics may improve the condition. Moving on to the adipocyte-myocyte islet axis. So adipokines are cytokines that are secreted from adipose tissue. And the most famous adipokine is leptin. And leptin is known to suppress insulin secretion and decrease insulin sensitivity. So potentially it can bring about insulin resistance. On the other hand, adiponectin is an insulin sensitive sensitizer. It increases insulin sensitivity, increases glucose uptake in muscle and liver, and higher adiponectin levels translate to better insulin function. There are other adipokines as well, but their roles have not been fully fleshed out yet. Myokines are, of course, cytokines that are released from muscle, and fibroblast growth factor 21 is another famous myokine, widely expressed protein, broad mode of action, enhances insulin sensitivity. It is viewed as a potential therapeutic target for diabetes and obesity. Interesting one here. Interleukin 6 is considered an adipokine and a myokine, cytoprotective to the pancreas, and it has effect on alpha cell proliferation, brings about glucagon secretion. Moving on to the liver islet axis. So there are some liver factors which have effects on the islet. They are hepatocyte nuclear factor 3 beta and hepatocyte nuclear factor 1 alpha, pivotal in pancreatic development, maintenance of beta cell function, and loss of HNF1 alpha, abolished insulin secretion. So this is the liver islet axis. Here I have a question. Do both hepatic glycogen and muscle glycogen contribute to blood glucose levels? Do they? Okay. So you remember this glycogenolysis, and here is this enzyme glycogen phosphorylase, which I said is present in muscle, brain, and the liver. And I said this enzyme glucose 6-phosphatase is present in substantial amounts only in the liver. So this last step for formation of free glucose can be done only in the liver, because glucose 6-phosphatase is present in substantial amounts only there. In the muscle, this enzyme is not there, so the reaction can come up to here and muscle can use glycogen, but it cannot release glycogen. So it does not contribute to systemic blood glucose levels. And now we move on to the brain islet axis. So there's a reciprocal relationship between the brain and the islets. The islets are richly innervated with sympathetic and parasympathetic nerves, and parasympathetic stimulation increases insulin secretion, and the converse with sympathetic stimulation. And there are insulin receptors which are widely distributed in the brain. In humans, we find them in the hypothalamus, in the hippocampus, in the cerebellum, and in the cerebral cortex. And in rat brains, they're found in even more areas. And it is also known that certain brain regions affect pancreatic hormone secretion. For example, the ventromedial hypothalamus, it has inhibitory effect on beta cell. So destruction of that region results in insulin hypersecretion. And the cephalic phase insulin response. This is a conditioned reflex where insulin is released even without nutrients or glucose. And it has been suggested that cholinergic and non-cholinergic processes, as well as the dorsal vagal complex in the medulla, are involved here, and it prepares the organism to adequately respond to incoming nutrients. Thus, we see that though the pancreas has a central role in glucose regulation, there is a lot of interplay with other organs and tissues, as we just saw. Before we move on to neural regulation, just some light relief. Okay. So now we'll move on to neural regulation of glucose homeostasis. So you might be surprised to know that way back in 1854, Claude Bernard reported that a lesion in the floor of the fourth ventricle in dogs altered glucose levels. 1854. Since then, little info came out on the neural regulation of glucose homeostasis, but we can't call it much. However, since 2010, quite an amount of info has been coming out on the neural regulation of glucose homeostasis. So what we are going to talk about now is how blood glucose is sensed by the brain, which regions of the brain are involved, what are the circuits involved, and how do they bring about actions on peripheral metabolism. Okay, this is what we're going to talk under neural regulation. So we know that the brain plays a vital role in glucose homeostasis. It has highly organized circuits in many parts, especially in the hypothalamus and the brainstem. There are different cell types that detect changes in glucose, and the brain also senses the peripheral metabolic state through signals such as insulin and leptin. So the regions involved in glucose homeostasis in the brain are mainly the hypothalamus and the brainstem. In the hypothalamus specifically, the arcuate nucleus, the ventromedial hypothalamus, paraventricular nucleus, and the lateral hypothalamus. And in the brainstem, we have the dorsal vagal complex, which is comprised of the NTS, the area postrema, and the dorsal motor nucleus of the vagus. So these are the regions that are involved in glucose homeostasis. Of special interest is the arcuate nucleus of the hypothalamus. You can see it over here. This is the arcuate nucleus. You can see that it's very close to the third ventricle, so a good position to assess glucose levels. And it receives multiple inputs from different parts of the body. And there are two sets of neurons in the arcuate nucleus, the POMC neurons, pro-opiomelanocortin neurons, which secrete alpha MSH, melanocyte stimulating hormone. And we have AGRP-NPY neurons, which secrete a Guti-related peptide, AGRP, and neuropeptide Y. And these neurons signal downstream through receptors, the melanocortin receptors and the neuropeptide Y receptors. Now, the hypothalamus receives multiple inputs from multiple parts of the body. So it's important that these multiple inputs are important because it allows for rapid response by the hypothalamus. So we have information from the stomach, from taste buds, from blood, intestinal mucosa, hepatoportal glucoreceptors, GI hormone signaling, adipose tissue signaling, signals from the cortex, wide range of signals coming into the hypothalamus. And glucose itself is an important afferent signal to the brain. Now, glucose crosses the blood-brain barrier and it enters through GLUT1 and GLUT3. And this entry is insulin independent. Insulin independent. So, what are the glucose sensing mechanisms in the brain? So, the glucose sensing neurons include glucose excitatory neurons, which respond to high levels of glucose, and glucose inhibitory neurons, which respond to low levels of glucose. So shown here is the mechanism of action in a glucose excitatory neuron. You can see here this glucose entering through GLUT234, taken up by glucokinase, converted to ATP. There's an alteration in the ATP-ADP ratio, acts on the ATP-dependent potassium channel to close it, brings about depolarization, voltage-dependent calcium channels open, and there's neurotransmitter release. So this is the mechanism by which GE neurons sense glucose levels. Coming to GI neurons, they also express GLUT234, they also have glucokinase, but we're not very sure about how these neurons act. It is suggested that glucose enters, is converted to ATP, bringing about alteration in ATP-AMP ratio, and then either by action on chloride channel or the sodium-potassium pump, hyperpolarization is brought about in the GI neuron. So where are these GE and GI neurons present? GE neurons are mainly present in the ventromedial hypothalamus and in the paraventricular nucleus. We know that these are the satiety areas of the brain. They are also present in the POMC neurons of the arcuate nucleus. What about the glucose inhibitory neurons? They are present in the lateral hypothalamus, which we know to be the feeding center. Interestingly, there are also non-neuronal glucose sensors, and they include glial cells such as the astrocytes, tanycytes, perivascular macrophages, and the microglia. So the astrocytes, are part of the blood-brain barrier. You can see an astrocyte here with its foot processes on the blood vessel. They're present at the interface between blood vessels and neurons. And this is an astrocyte. This is a neuron over here. Glucose enters the astrocyte through GLUT1. It is processed to pyruvate and then to lactate. And then the lactate is shuttled through the monocarboxylate transporter to the nearby neuron, where it is converted to ATP, leads to neurotransmitter release and signaling. This is how glucose sensing takes place in the hypothalamus. Okay, glucose sensing in astrocytes. This is what works in the hypothalamus. Now coming to tannocytes. These are specialized ependymal cells surrounding the third ventricle. Third ventricle is here, and these cells over here, these are the tannocytes. You can see that they have processes which extend into nearby neurons. So here, this is the mechanism of glucose sensing. Glucose enters through GLUT2, again glucokinase, converted to lactate, transported through the monocarboxylate transporter, converted to ATP, acts on the ATP-dependent potassium channels, voltage-dependent calcium channels, open to let in calcium and neurotransmitter release. So this is how glucose sensing takes place in tannocytes. This also is important in the hypothalamus. And thirdly, there are what are called the perivascular macrophages. Now, these are present in the fluid-filled space surrounding small arteries in the brain. It's called the Virchow-Robin space. And they sense glucose flux from the circulation to the brain. And this is critical for adequate glucose supply to CNS neurons in high-fat diet conditions. So when there's going to be excess of fat, still adequate glucose has to be supplied to the brain, and that is where these perivascular macrophages play a role. Take a look at this picture. It shows the PVM's role in a high-fat diet. So the first picture shows a normal diet. You can see a blood vessel here, endothelial cells here, perivascular macrophages here, and what you see here are astrocyte food processes, and these are hypothalamic neuron. Okay. So normal diet, glucose molecules shown in green, lots of glucose is there, getting into the hypothalamus through GLUT1 transporters. Come to the next picture. After high-fat diet of 3 days, what we see is there are more fat particles in the circulation, and the amount of glucose getting into the hypothalamus is decreased. Move to the third picture. After high-fat diet of 28 days, now you can see there's a lot of fat in the circulation. But what has happened is the PVMs, the perivascular macrophages, have secreted VEGF, vascular endothelial growth factor. That leads to an upregulation of GLUT1 receptors, and because of that, there is restored glucose flux to the hypothalamus. This is the important role of the perivascular macrophages in a high-fat diet. Under conditions of reduced glucose flux, PVM secrete vascular endothelial growth factor, which restores the glucose flux by increasing GLUT1 expression. And the last kind of cell is the microglia. These are situated near NPY neurons. We know they are brain-resident immune cells. They're there in other areas also. And importantly, they sense fluctuations in glucose and regulate neuronal responses. So these are the glucose sensors in the brain, neuronal as well as non-neuronal. So now we'll move on to neural circuits that control glucose homeostasis. So quite a lot of recent work has gone on in this regard, and a few circuits have been established. So we are going to take a look at those now. We have just 5 of those. So there's this first circuit which involves the ARC, AGRP, and PY neurons, arcuate nucleus, agouti-related peptide, neuropeptide Y neurons, and you must be wondering what this structure is. This is the bed nucleus of the stria terminalis. It's a limbic forebrain structure. It's part of this circuit, the anterior bed nucleus of the stria terminalis. So we have AGRP neurons projecting to the ABNST, and from there through the vagus and the sympathetic acting on peripheral metabolism. So through the vagus increases HGP, and through the sympathetic decreases glucose uptake by cells. So this pathway, this circuit decreases glucose tolerance. The next circuit I'd like to talk to you about is the arcuate nucleus pro-opiomelanocortin circuit. So here, POMC neurons project to the paraventricular hypothalamus, where there are melanocortin receptors, and then again through the ANS, they bring about upregulation of GLUT2 receptors in the kidney and increase glucose reabsorption. Thus, they raise blood glucose level. And the third circuit is going to be the steroidogenic factor 1 neurons of the ventromedial hypothalamus. Here there are 2 effects through the ABNST. Through the ABNST, it acts to increase HGP and increase blood glucose level. Through the central melanocortin signaling here, through that, it acts to increase insulin sensitivity. So basically what we're trying to say is there are different circuits bringing about different actions on different peripheral organs. Lateral hypothalamus, we have two kinds of neurons, the glutamatergic and the orexin producing neurons. They act through the sympathetic to increase glucose uptake. And lastly, there is a circuit which involves the brainstem dorsal vagal complex. As we mentioned earlier, the dorsal vagal complex includes the NTS, the DMV, and also the area postrema, and it's crucial for sensing and regulating glucose fluctuation. And a range of actions here, depending on the neuron. We have NTS GABAergic neurons, which sense hypoglycemia and trigger glucagon secretion. Brainstem melanocortin signaling is important in the central regulation of glucose metabolism, and we have a role in satiety being played by NPY, POMC, and GLP-1 neurons of the brainstem, TVC. And now moving on to central signaling within the brain. So we have a few important signals that we'll take a look at now: insulin, leptin, and glucagon. So insulin receptor, we already saw, is expressed in many brain regions. We said cortex, cerebellum, the hypothalamus, hippocampus, and it has effects on peripheral glucose metabolism. It suppresses hepatic glucose production and reduces expression of gluconeogenic genes. And this action is through the arcuate nucleus, AGRP neurons, via the parasympathetic nervous system. So studies in humans have shown that intranasal or intracerebroventricular insulin infusion decreases peripheral insulin sensitivity. So it's postulated that insulin may stimulate glucose uptake by bringing about AGRP inhibition. Interestingly, insulin acts on the brain to influence its own secretion and also the secretion of the counter-regulatory hormones. So ablation of hypothalamic GLUT4 expressing neurons has been found to decrease insulin secretion, and brain insulin signaling has to be proper for the hypoglycemia counter response as well. And the post-meal insulin response. So after meal, insulin enters the brain via the blood-brain barrier, and it stimulates POMC neurons, inhibits AGRP neurons. So stimulates POMC neurons to decrease food intake, inhibits AGRP neurons, which are orexigenic, and this is pivotal for normal postprandial glucose management. So this is the important point. Both peripheral and central insulin signaling are impaired in obese or diabetic states. Dysregulated insulin signaling has been implicated in overnutrition conditions, and it causes adverse metabolic outcomes in certain subsets of neurons. For example, in POMC neurons, dysregulated insulin signaling results in insulin resistance and increased hepatic glucose production. In the LH, it leads to hyperinsulinemia. In the VMH, it promotes obesity and glucose dysregulation. Thus, central insulin signaling has to be in proper form. If not, it leads to disease states. Next, we come to central leptin signaling. Now, leptin, is secreted from adipose tissue, important signal from the periphery to the center, and it acts through the ARC-POMC neurons to inhibit food intake, important modulator of peripheral glucose metabolism, suppresses insulin secretion, decreases insulin sensitivity, potentially leading to insulin resistance. It's a known fact that in obesity, leptin levels are highly raised. And then central signaling by GLP-1 and glucagon. So GLP-1 has effects through the hypothalamus to bring about increase in insulin secretion and to decrease hepatic glucose production. So that's the central signaling by GLP-1. Central signaling by glucagon. This is interesting. So central signaling by glucagon results in decreased hepatic glucose production. We know that peripherally glucagon acts to increase hepatic glucose production. So probably there's a negative feedback loop that operates here to fine-tune the secretion of glucagon. So we have seen that the pancreas plays a central role in glucose homeostasis by its action on different organs and tissues. And blood glucose levels are sensed by peripheral glucose sensors and also by central glucose sensors. Central sensors include neuronal sensing and non-neuronal sensing. Neuronal, we spoke about the GE, glucose excitatory, and the glucose inhibitory neurons. Neuronal, we spoke about four kinds of cells which participate, namely the astrocytes, the tanocytes, the perivascular macrophages, and the microglia. And then we took a look at the efferent glucoregulatory circuits. We spoke about 5 circuits and their impact on peripheral glucose metabolism. So this is neural regulation in a nutshell. Now, an interesting thing about the link between obesity and diabetes and how it's there in the brain. So what is shown here, you can see a blood vessel, you can see endothelial cells. What are here are the perivascular macrophages. You can see some microglia here, astrocytes here, neurons over here in the arcuate nucleus. So in high-fat diet and overnutrition states, what happens is these glial cells get activated to produce pro-inflammatory cytokines. They're activated to produce pro-inflammatory cytokines. And these activate inflammatory signaling pathways, which lead to impaired leptin and insulin signaling and dysregulated glucose metabolism. Additionally, activated perivascular macrophages, what they do is they act on inducible enosynthase, okay, which is a vasodilator. So it makes the blood-brain barrier more permeable, which means these fat molecules get into the hypothalamus, and that leads to inflammation in the hypothalamus. And this is obesity-induced hypothalamic inflammation. So this is the link between obesity and dysregulated glucose metabolism in the central nervous system. So a blockade of inducible enosynthase in hypothalamic PVMs was found to significantly improve systemic insulin resistance and glucose intolerance in high-fat diet-fed obese mice. So it is suggested that INOS signaling in hypothalamic PVMs may regulate systemic glucose metabolism and insulin resistance via as yet unclear mechanisms. So this is just to say that insulin resistance and glucose intolerance, the center is in the CNS. So in health, the well-functioning interactions between all of the organs and tissues involved ensure glucose homeostasis. But impairments in the secretion of and or sensitivity to insulin lead to metabolic diseases such as type 2 diabetes mellitus. Okay? So a quick look at diabetes and we are done. So what is diabetes? It's a syndrome of impaired carbohydrate, fat, and protein metabolism caused by a deficiency of the effects of insulin at the tissue level. So types of diabetes, we know there's type 1, type 2, and also secondary diabetes. Type 1, where there's hardly any insulin production. Type 2, where there's insulin resistance, hyperglycemia, and a relative insulin deficiency. And secondary diabetes, which is due to other endocrine causes. As you can see, type 1 and secondary are a smaller percentage. The bulk of diabetics have type 2 diabetes mellitus. And in India, it's a serious problem. It seems because of the Asian Indian phenotype, where the big belly and thin extremities are there. So it's of great importance in our country. And type 2 diabetes is commonly preceded either by obesity or by insulin resistance, or even by metabolic syndrome or syndrome X, which has all these components: insulin resistance, fasting hypoglycemia, hypertension, dyslipidemia, and obesity. Now, the basic fundamental biochemical defects in diabetes are the following: increased hepatic glucose production, decreased tissue glucose uptake, decreased amino acid entry into muscle, and increased lipolysis. The first two being attributed to impaired carbohydrate metabolism. So there's increased hepatic glucose production and decreased glucose uptake by cells, resulting in hyperglycemia and intracellular glucose deficiency. So what are the effects of hyperglycemia? So take a look at the picture. So there's an increased glucose load being delivered to the kidney. Some amount is absorbed, threshold is exceeded. Therefore, there's going to be glucose in the urine, which is called glycosuria. Because of the increased glucose load coming to the kidney, water is being retained in the lumen, there's going to be increased urine flow. This is the cause for the polyuria in diabetes. Because of the fluid that is being lost in urine, there's going to be hyperosmolality of the blood. And this hyperosmolality draws water from the cell, leads to intracellular dehydration. There's also urinary loss of sodium and potassium. And the effects of intracellular glucose deficiency, less glucose utilization in cells because there's hardly any glucose inside the cell despite the large amount of glucose in the blood, poverty in the midst of plenty. So the energy needs of the cell are met by protein and fat catabolism, and that leads to ketosis. Deficient hormone sensing in the CNS, that's the cause for the hyperphagia. And coming to cholesterol metabolism, excess fat utilization by the liver over a long period results in high levels of cholesterol in the plasma, which get deposited in blood vessels, and it accelerates atherosclerosis. Excess acetyl-CoA in the cells is converted to ketone bodies, acetoacetate, acetone, and beta-hydroxybutyrate. These remain in circulation, and in association with dehydration, they lead to acidosis. And we said sodium and potassium are being lost in the urine along with water. That leads to dehydration, hypovolemia, can lead to hypotension, coma, and death. Okay? So additionally, there are changes in protein metabolism also in diabetes. Catabolism of protein increases. There's protein depletion, muscle wasting, asthenia and weight loss, changes in fat metabolism, catabolism increases, synthesis decreases, system is flooded with triglycerides and free fatty acids. One important complication of diabetes is hypoglycemia. We know that when values go below 80 milligrams per deciliter, we call it hypoglycemia. And when the value is at 70, the CNS becomes excitable, and ANS features such as sweating, tremors, and nervousness are manifested. Now, this serves as a warning sign for an impending hypoglycemia. But there is this condition which is called hypoglycemia unawareness, which occurs especially when there have been repeated bouts of hypoglycemia. And in hypoglycemia unawareness, these autonomic components become missing, which means the person has no warning of an impending hypoglycemia. It's potentially dangerous, can lead to death. And the complications of diabetes, chronic high glucose concentration causes tissue injury. So we have vascular complications, macrovascular, microvascular, and we also have neuropathy. Macrovascular complications are brought about by the atherosclerosis, and microvascular complications and neuropathy because of the chronic hyperglycemia. Aldose reductase enzyme is activated and lots of glucose is converted to sorbitol. The sorbitol accumulation leads to the formation of amidoride products that form AGEs and damage the blood vessels. So this is the cause of microvascular complications and neuropathy in diabetes. I'm almost at the end here now. So moving to the management of diabetes, type 1 we know usually managed with insulin, but for type 2, diet and exercise generally can work at the earlier stages, but over time most patients progress to needing drugs. So therapeutic agents, the modern day practitioner has an arsenal of drugs that he can use in the fight against diabetes. I would like to draw your attention particularly to the incretin mimetics over here. Incretin mimetics, relatively newer drugs. They are GLP-1 receptor agonists, exenatide, liraglutide, and semaglutide. And the important thing about these is that the hypoglycemia that is seen commonly with sulfonylureas is not seen with these incretin mimetics. Certain other molecules that have come newer are the gliptins. Now, the gliptins are DPP4 inhibitors. DPP4 is the enzyme that inactivates the incretins. So, these inhibitors slow down the breakdown of incretin hormones. They include drugs such as sitagliptin and saxagliptin. Then we have the gliflozins, which reduce glucose reabsorption in the kidneys by inhibiting sodium glucose transporter 2 in the kidneys. And lastly, we have the amylin mimetics, example pramlintide. They reduce glucagon secretion and they slow down gastric emptying. And it seems insulin and pramlintide injected separately, both before a meal, work together to control the postprandial glucose excursion. And just to look at some newer agents that are in use now. So to summarize what we have seen today, we spoke about the importance of blood glucose regulation, how it's the only substrate for brain and therefore levels cannot be too low, how if it's too high, it leads to tissue damage. So we have mechanisms of regulation, which include the liver, which is the glucose buffer system, and then we have the pancreatic hormones, and their interplay with other tissues was discussed, the intra-islet insulin axis, the gut islet axis, the adipocyte myocyte islet axis, liver islet axis, and the brain islet axis. And lastly, we spoke about the CNS role in glucose homeostasis. We spoke about neuronal and non-neuronal glucose sensors, neural circuits, and the central signaling that goes on in the brain. So though we appear to have made great strides in our knowledge of glucose homeostasis, our knowledge, especially of the neural regulation, still remains in infancy. Further research is needed to identify new therapeutic targets in our fight against diabetes. So in closing, I thought today being World Poetry Day, I should close with a kural. தீயளவன்றி தெரியான் பெரிதொன்னின் நோய் அளவன்றி படும். And here is Reverend Dr. G. Pope's translation of the same, who largely feeds nor measure of the fire within maintains, that thoughtless man shall feel unmeasured pains. So immoderate eating should not be there, that person will land up in a lot of trouble. That's a simplification of the Quran. So that brings me to the end of my presentation. Thank you very much. Any questions?
Thank you so much, Dr. Preetha Paul, ma'am, for this insightful and enlightening section. Your deep dive into glucose homeostasis and its physiological impact has truly enhanced our understanding of this critical process. Now we'd like to open the floor for any questions. We'd like to invite questions from the audience. If you have anything like you would like to discuss further or seek clarification on, please feel free to raise your hand or type your questions in the chat box. Now I request Dr. K. Divyalakshmi ma'am to deliver the vote of thanks.
Any questions? I have just this. First of all, it was a wonderful presentation by you, a great lecture, and I think we were all quietly struck.
And you gently escalated the flow of the presentation. So thank you so much, Preetha. It was a joy listening to you. I noted down a few things for me to go back and read. But when you spoke about the short chain fatty acids and they reduce the insulin secretion, you also told that the long chain fatty acids increase the insulin secretion. Very recently, when we were reading a little about the microbiota, so we were talking about consuming diet that can increase the short chain fatty acid production in the gut. So, and they say that when the short chain fatty acid production is increased in the gut, we derive a lot of benefits. And so when you say short chain fatty acids reduce the insulin secretion, I was, I thought maybe I should go and read a little bit about, because if it reduces the insulin secretion, will it affect the glucose metabolism? This is at the gut level that we are talking, and I'm not sure how much it will actually influence the glucose homeostasis.
This is a relatively newer finding, and studies have shown that the long-chain fatty acids stimulate insulin secretion and the short-chain decrease. Probably there are other factors that also operate. So we do not know the interactions that are there. We are just looking at it from a simplistic point of view. So maybe we need to go deeper into it to work out this point that you're bringing out.
Sure, Preetha. And then tanny sites was something new to me. I've never heard about tanny sites. And as you spoke, I just quietly browsed to see what it is. So, is it another neuroglial cell, or is it along with the glial cells,
It's
the
ependymal cell, which lines the third ventricle.
ventricle? Yeah, yeah.
And there are four types of tanycytes actually: alpha 1, alpha 2, beta 1, and beta 2. And alpha 1, alpha 2, and beta 1, they mediate transport between the CCs and glucon. And beta 2 alone mediates transport of, for example, leptin from the blood vessel to the neuron. And
And the way you showed those three pictures about high fat diet, that was so beautiful when you spoke about how the vascular endothelial factor after 28 days it upregulation. So should I understand, or am I correct in understanding that people who adopt a high fat diet, mm, they initially think that it's helpful, right? You know, the blood glucose levels. People generally have this misconception that when you go on a high fat diet, they're replacing glucose with fat. But at the end of the 28 days, if everything is brought back to normal by somebody else, then the role of high fat diet itself becomes very
but then we saw
questionable.
another one where we saw how high fat diet leads to hypothalamic inflammation.
Yeah, yeah.
That's the other
So
side of the coin.
it isn't
So
advisable in
probably
this
the upregulation of glucon is a homeostatic mechanism trying to bring it back to
bring it
normal.
back to normal. Yeah.
But the damage is done when a person is on a high fat diet.
Because generally, basically, any sort of fat diets like this, where one macronutrient overtakes the other, it is not advisable. And I think that picture was a beautiful way to show us that even though initially you might see a little bit of changes, with 28 days, the system puts us back in place, and one shouldn't be probably taking up these.
That's the beauty of the human body, right? So many corrective mechanisms which the good Lord has put in place. Somehow, all the damage that we do, there are certain homeostatic mechanisms that try to bring it back to normal. But then, when sustained, the body cannot handle those insults.
You've set us into a lot of thinking, Preetha, and the way you did the circuits, the inter circuits and the neuronal circuits, I think it was simply amazing. Thank you for sharing the references. I hope our postgraduates pick it up and dive into it. Thank you so much, Preetha. Thank you. Right.
Yes, thank you, ma'am, for the very uh, evidence-based and especially intense intense treatment on the neuronal regulation. Basically, we are more convenient and comfortable with endocrine regulation, but when it has come to neuronal regulation today, I've seen so much new things, so many good things I didn't know, all the inflammatory effect of high-fat diet, all those things. So thank you for your good presentation, ma'am. I have two things. Now it's the season in which Muslims and Christians are fasting, and you spoke of ghrelin, the hunger hormone. Probably they are having this ghrelin in a high amount. So you said ghrelin is cytoprotective. So does it protect only the beta cells of the Langerhans, or this cytoprotective effect of ghrelin is to all body cells?
Cytoprotective effect is?
See, you mentioned ghrelin as
Is it only for beta cells or?
is it only for beta cells of the pancreas, or it is cytoprotective for all other cells?
No. Specifically for the pancreas alone, for the beta cells, yes.
Okay, ma'am. Thank you. Another question is about you spoke of the effects of exercise and stress on how are they influencing glucose utilization. Now, if we have been having a World Sleep Day events past few weeks. So, is there any role of circadian system? Then the role of circadian system in glucose homeostasis, and how does this circadian rhythm affect glucose? Could there be any effect on circadian rhythm apart from exercise and stress influences, which you have elaborated well in your presentation?
Very interesting question. And as I was researching this topic, I came upon a paper which speaks about a circadian rhythm in glucose homeostasis. But I didn't go too much into it, so I'm afraid I can't give you the details. But I can tell you that definitely there is a circadian system operating. And even something about the astroglial cells and their activity and the circadian rhythm operating there as well. So that's the only thing I can tell you for now.
We see that there's, as much as we talk about the suprachiasmatic nucleus
Yes.
and the pineal gland as a central clock, they say there are peripheral clocks. The peripheral clocks, of which there is one gut clock, there is one pancreatic clock, and so they have a little circadian influence on the glucose levels. So, like Preetha, I also very, very little bit looked into it when I had a lecture on sleep and gut. So that is when I realized that there is a peripheral gut clock and pancreatic clock, which also operates in this.
All these are newer things which we must read about a little more. But that's a lovely question. Thank you.
Sleeplessness, I think, may also indirectly affect the glucose homeostasis because sleep again leads you going for stress. Stress.
And metabolism is affected. Yes, ma'am. Yes. Definitely that also.
Presentation, Preetha. Actually, so proud of you.
Thank you,
It's so
ma'am.
nice to see our own student performing so well. It was really fantastic performance.
Thank you so much, ma'am.
So, started it in a very simple way, then slowly actually said you raise the bar. That was it. One question.
Yes, ma'am.
It's a keto diet.
Ah.
What's your suggestion?
Ma'am, then again, disclaimer, I have not done great study on these diets, but personally for me as a doctor, I feel moderation is the key. I like what the squirrel says. Moderation is the key for
Our students can also fill up the post test and feedback form. Now, when you freshly remember everything about would you like to give a closing in the book? Or?
Presenting the PALS post-ordinary.
Yeah, sure. There's one big question. The PDF of the lecture can be shared. It's got one participant has asked.
Okay.
If you permit, it's it. So thank you everyone for your patient listening and thank you for the questions which show that you really listened. Thank you very much for having me here.
Now I request Dr. K. Divyalakshmi ma'am to deliver the vote of thanks.
Thank you, Jamie. A very good morning to everyone who are with us online and offline. It is indeed a great privilege for me to deliver the vote of thanks for this wonderful occasion. Let me begin by expressing my heartfelt gratitude to the management officials, our Honorable Chancellor, Pro-Chancellor, Vice-Chancellor, and Pro-Vice-Chancellor for providing us with state-of-the-art infrastructure and for the unwavering support in all our academic activities. We extend our sincere thanks to Dr. Balaji Singh, Dean of Medical College, for being the constant pillar of support. And our heartfelt thanks to Dr. K.V. Swaminathan, sir, for graciously joining us and also facilitating our guest lecture.
Our sincere gratitude goes to our distinguished guest speaker, Dr. Preetha Paul ma'am, for graciously taking time from her busy schedule to share her knowledge and expertise with us. Thank you, ma'am. A special thanks to our wonderful alumni who continues to a continued connection and contribution plays a vital role in inspiring and guiding our students. A special mention to Dr. Bindu ma'am for joining us today and our alumni, Lavanya and Priyadarshini for joining us today. It is my pleasure to extend my heartfelt thanks to our head of the department, Dr. Muthu Pratibha ma'am, for organizing and hosting such great guest lectures that stimulate higher order learning among the undergraduate and postgraduate students. We are grateful to our senior professors, Dr. Padmavati ma'am and Dr. Priscilla ma'am for their continuous encouragement. A special vote of thanks to Dr. Meenakshi ma'am, our PALS 3 team coordinator, for her sincere effort in planning and coordinating today's program. I would also like to thank Pavitra, second year MD degree, for preparing the invitation, Mr. Alan and Dr. Vivek for co-coordinating with Dr. Meenakshi ma'am and for managing the audiovisual arrangements, and Ms. Jemmy for anchoring the event. Thanks to Ms. Maya and Tamal for beautifully capturing today's event. I extend my sincere appreciation to all the faculty members, technicians, Secretary Ms. Padma Purvi, and support staff of the Department of Physiology for their tireless effort in making this program a grand success. Last but not least, we also thank all the participants, both online and offline, for joining us today. We hope this session is educative and enlightening, especially on the core concept of glucose homeostasis. We also thank our communication and audiovisual team for their continuous support in enabling us to conduct this program in hybrid mode smoothly. Thank you all once again. Wishing you a very wonderful day. See you all in PALS 4.
As we come close to this insightful session, I want to sincerely thank Dr. Preetha Paul ma'am for her time, expertise, and invaluable contribution for today's session on glucose homeostasis. A big thanks to the Department of Physiology for organizing this initiative and to all the participants for your enthusiasm and engagement. Your presence and interaction had made PALS a vibrant platform for learning and knowledge sharing. We hope today's session has inspired new ideas and reinforcement the importance of staying connected with our alumni network. Let us continue to learn, collaborate, and continue to the growth of physiology as a field. Before we wrap up, a quick reminder, e-certificates will be provided to all the registered participants only if you fill the post-test and feedback forms that's posted in the chat box. Stay tuned for the upcoming PALS lectures. Thank you once again, and we look forward to seeing you in the next session. Have a great day ahead. Thank you.